appreciate it, and I know everybody else does too. Okay, good afternoon, everyone. Welcome to our 1 o'clock briefing today, Living Donor Tissue Propels Brain Research. We have two speakers in our briefing today. They are Ed Lein, the Director of Human Cell Types Program at Allen Institute for Brain Science here in Seattle, and Karen Rommelfanger, the Director of Neuroethics Program and the Professor of Neurology, Psychiatry, and Behavioral Sciences at Emory University in Atlanta. We'll go ahead and get started with their remarks, and then we'll move to Q&A. Thank you.
We just talk from here. Okay. Okay, thank you very much for the kind introduction. What I'd like to tell you a little bit about today are advances in human brain mapping that we've been making and the importance of actually soliciting living donors to contribute to this research. As many of you probably know, for many years now we've been engaged in trying to map the fine structure of the brain using genes, using anatomy at the Allen Institute for Brain Science, and using these to make sort of catalytic public resources to push the whole community forward. In recent years, huge advances have been made, particularly in model organisms, at mapping the cellular architecture of the brain and to try to treat the brain as a very complex cellular organ that can be dissected down to a finite number of cell types. One particular driving technology for this is coming from genomics to use a technique called single-cell transcriptomics to measure all of the genes in individual cells. This technology has really sort of exploded in the profiling of very complex brain tissues in mouse and in monkey, and we've been among the first to try to apply this now to the human brain. Doing these types of studies, of course, requires voluntary donations of autopsy specimens. And using this technique, we've been able to show that in fact the brain is about as complex as everyone thought it was. It's extremely cellularly complex, but at the same time finite and definable. And using this sort of technique, we can sort of bound this problem of thinking of the brain as made up of a whole series of these definable circuit elements. We had a recent paper in the journal Nature on this that you can look up. And also in this study, we first did a comparison across species to see how well conserved is this architecture from human to mouse that we might like to use as a model organism to study human disease. And the answer is a bit complex. On the one hand, things are really quite similar. The overall architecture is quite conserved, but on the other, the details of those types of cells vary significantly across species, including for things like receptors that could be drug targets for treating disease. As we kind of get this framework of cell types, a big area of focus recently is trying to move from studying what we can do in a post-mortem dead brain to actual functional dynamic properties of cells in living tissues. This, of course, proposes a pretty significant challenge because we have to have access to living tissues. But we managed to establish a program to work with local surgeons to have access to neurosurgically resected pieces of tissue that are normally taken out in the course of epilepsy treatment surgeries or the removal of deep tumors. And it turns out that these tissues are extremely robust. They're highly consistent despite coming from people with disease. And we can apply many of the most recent tools used to study brain architecture in model organisms to study the human brain. One of the things that we can do with this is to look at the fine anatomy of individual cells. This picture up on the screen here is one example of a dye-filled cell. You can see how complex these cells are. Normally you think of a cell as a little cell body. In fact, neurons have extremely complex processes, as well as the physiology of the cells, the electrical properties of the cells, and the genes that they use, all in these living tissues. In fact, they're so robust that we can even keep these alive for longer periods of time, which allows us to develop genetic tools to target particular types, which is actually a pretty clear path to gene therapy applications to do very precision medicine. As we start to get this data, and we'll have many publications coming from this in the coming years, we're really seeing that when you look at this fine granular level of detail, there are many species differences. There are differences in the variety of cell types, how many types of cells there are, specialized properties between species. as well as differences in gene expression for really functionally important genes, I might add, that are not used in the same kinds of cells. And so these are really kind of revealing, I think, the nature of a paradox, which is an animal like a mouse is sort of deceptively similar. We would like to use it to study human disease and to develop cures for human disease, but many of the details actually vary significantly. And in many cases, these can be really profound differences that have functional implications and that make it quite difficult to use that as a model. So while model organisms have enormous utility in the field, it's really useful, it's necessary, I would say, to understand what you're trying to model. And there's no real substitute at the moment, even with things like organoids or stem cells, than the actual living human brain to study this. So these types of approaches are making really quick advances, and I'll be sharing that in my talk. Hopefully all of you can make that, and I'm happy to answer any questions.
Okay, good afternoon. I'm going to make 3 main points today, and I'm going to keep it short so we have some time for discussion. The first is that I want to emphasize that patients want to donate their tissue. And if they want to, they should be able to. So in study after study, patients who are in a position to provide tissue from clinical procedures have repeatedly stated that they'd like to donate their biospecimens to research. They feel it's a way for their illness to help others, and as long as it isn't used to make a profit, they're generally supportive. This research is truly remarkable, presaging remarkable neuroscience insights for society, and it's a spectacular opportunity for patients to once again get involved in frontier science. And if they want to volunteer to contribute in this way, then they should be able to. My second point, patients need to truly volunteer. Volunteerism is what prevents humans from being reduced to a standing reserve of tissue, organs, and body parts. The scientific process in this one, and any value that this kind of research could provide, is completely undermined if even one human is treated unjustly. Good science is ethical science. And in order to truly volunteer, would-be participants need to know the risks and have some authority over how their donation is going to be used. First, they need to know how that tissue or donation is going to be used in the future. For example, some participants may not want skin tissue used to create brain organoids that might eventually develop some kind of spontaneous electrical activity, as far as that might actually be from consciousness, because to date, nothing like that exists. Second, they need to know about how the data is going to be used, and perhaps more importantly, the possibility of re-identification, which these days seems to be an impossible likelihood, or a impossible unlikelihood, I suppose. And in the wrong hands, this could contribute to stigma and discrimination. My third point is this. Innovation in ethics and its tools needs to keep up with the science. The onus of addressing these ethical issues cannot be on scientists alone. The onus is on ethicists as well. With the evolving science and the increasing global nature of science, we need to make sure our thinking about ethics and the tools we use for ethics are still relevant and useful. First, ethics needs to keep up with the global nature of science, considering the global exchange of materials and findings. I co-lead something called the Global Neuroethics Summit. I guess I should use this now. Oh, maybe not. You don't have, that's fine. You don't need it. I will tell you what it is. I co-lead something called the Global Neuroethics Summit. This is a product of a global neuroethics working group of the International Brain Initiative. The International Brain Initiative is a consortium of 7 large-scale existing and emerging national level research projects in neuroscience around the globe, and their total investment to date, their total investment plans exceed $7 billion. And there's a paper that just came out a couple weeks ago in Neuron introducing that entity. Our global neuroethics group has existed for a while, and one of the first things that we wanted to do was to bring the neuroethics components of all the brain research projects to talk about ethical issues and analyze them through a cross-cultural lens. One of the first things we did was create a list of 5 neuroethics questions for neuroscientists to ask, and I wanted to have those up for you, but they are published in Neuron a couple times. In 2018 was their first release. In 2019, the first special issue in Neuroethics was released where all the brain projects talked about how they use them. But I'll draw your attention to the second and third of those questions. The second question invites scientists to explore how the standards of data collection and specimen collection and how those might vary across the globe. The third is a question about the moral significance of the tissue that is being used and how that might be culturally explored. The second piece that I want to mention about innovation and ethics tools is that we need to make sure that they're up to date. We need to make sure like ethics instruments such as informed consent are not empty ethics consent tools. Informed consent is our primary tool for ensuring voluntariness for study participants. In an attempt to upgrade, recently the common rule, which guides federal human subject research, has seen several revisions, which I'm sure many of you know about. One revision here is the adoption of broad consent. Broad consent allows participants to consent to unspecified future research with their de-identified tissue. There's no need to reconsent when new studies beyond the original research plan to use the biospecimens. And this simply might not suffice with current living donor brain research when considering future uses of specimens and future data management. In closing, finally, what I hope for this press briefing and even my talk tomorrow is that we'll be able to empower future participants to have an increasing ability to engage with these types of science and society issues so that they can better inform the aspirations and guardrails for these types of research. Thank you.
Great, thanks. So we can take questions now, and if you raise your hand, a microphone will be brought to you. Kindly state your name and affiliation before your question. We'll go here, and then come here.
Tom Keenan from Business Edge. Ed, you mentioned possible drug targets in the future. I guess this question falls between the two of you. We know that with cancer patients, there have been cases of people whose tissue led to profitable drugs and they got nothing from it. Are you contemplating the financial side of this down the line? And that sounds incredibly complex as the brain. How are you going to do that?
I should say, our interests are in trying to catalyze progress as a whole. We are simply describing, and others can use that information. I do think it's an important and historical question of people's tissues being used to make significant advances without any benefit to them. I think this is something probably a little bit better in your court on this. We try to live in the open science and to describe these things so that people can maximally use this information, be it academic or commercial.
Thanks for that question. So your question was about commercial use of living donor tissue specimens. So generally, participants who have been interviewed and studied about this in many, many studies who are donating tissue for research do not want their tissue to be used for profit. They just don't. And I think with cutting edge research like these, that necessarily upfront, as those possibilities become available for monetization, that the participants need to go get consulted on that. And so that has to be a first step. And from there, it can be decisions about how profit should or should not be shared. And it doesn't mean that profit. Yeah. I understand what you're saying. What if you can't find those participants afterwards? There's a significant burden in trying to find those patients, right? And this is what drove the adoption of broad consent, because it's just too onerous to try to go back and tackle all those. But these studies are actually new enough so that we can bake in early on to start capturing these views. This is how ethics actually can be integrated into research design. During a consent process or a follow-up to these studies, you can ask participants about their views and perspectives on how this should be used and get inspiration and ideas on how you might navigate any kind of monetization from them. And importantly, as part of the scientific design, a lot of this is like organoids, for instance. People are thinking of using that type of stuff for personalized medicine. And right now it's so early that if you were to try to monetize it and apply it globally, it wouldn't actually be, unlikely to generate actual benefit for those individual patients who gave it.
Question here.
Thank you. Clive Cookson from the Financial Times in London. You've both briefly mentioned brain organoids. I'd be interested, Ed, in knowing what you could do with samples taken from patients that you can't do with brain organoids, which are, after all, living human brain tissue, and their proponents seem to think that they might be better than the sort of tissues you're working with. And perhaps you could go on to say how you're growing the human tissue, how that differs from organoids.
Yeah, it's a great question and a very important one right now because there's such a debate about what should be good models for studying human. The concept of an organoid is a perfectly legitimate one in that you have cells that came from a human being or even a particular individual that have the genetic program to become anything in the body. And if one could simply get that process rolling, you could generate those types of cells. In practice, it's very difficult to get these to look anything like the actual mature system. And a problem in the field right now is that really these systems can only be pushed through the very first stages of development. So if you actually want to understand a complex part of the nervous system, which has gone through 50 plus years of development and has long processes and connections with many cells in other parts of the brain, you simply aren't going to be able to do that with that kind of system, probably ever. That being said, I think there are many elements of these early stages which can actually be well studied, and there are lots of examples now of studies coming from diseased individuals where there are cellular deficits that you can actually study. So I think, again, I think it's a bit dangerous to start with something you don't understand and assume that that's a model. You should instead try to understand the system and then develop a model that recapitulates that allows you the benefits of a model, namely to do experimental perturbation.
My thoughts on the ethical differences of using living donor tissue like resected from the brain versus creating an organoid. Well, I think that in both cases, for participants, there is a certain, I don't know that there is a difference in that the tissue itself has a intuitive meaning or a cultural meaning where people identify with brain tissue regardless of whether it's as complicated as or as sophisticated as what Ed is doing or whether it's an organoid. I think what perhaps ethically is a little bit more nerve-wracking for people is that recently organoids were shown to develop spontaneous electrical activity. And certainly that spontaneous electrical activity has nothing that resembled that of a premature baby, or that's at least how it was highlighted in the news. That said, even that electrical activity didn't necessarily resemble what it would be like for a human breathing in society. But still, that creates enough concern for people that some individuals have stopped consenting to studies that use organoid tissue.
Question here?
Hi, Lee Holtz at the Wall Street Journal. Ed, I wonder if you'd just describe for us the processes that you have in place now for donation, collection, and consent for these tissues, and then the sorts of things that you're doing with them at the Allen Institute.
Sure. So I should say out of the gate that we don't manage the consent. We are recipients of this tissue. These are patients of local surgeons at the University of Washington and at Seattle Children's Hospital, Swedish Hospital, who are undergoing treatment either for intractable epilepsy or to remove a tumor. And so this is sort of really nothing unusual in these surgeries that's being done. In order to reach the site of pathology, it's necessary to resect a more distal piece of tissue. So we have simply established relationships to use these non-diagnostic tissues rather than being put in the waste. We can actually collect them and try to use them for doing these types of studies. In practice, we have a team that will go to the hospital and receive these tissues outside of the clinic and immediately take them to be worked with in the laboratory. This has to be done quite quickly. These are labile tissues that if they're not treated carefully and processed quickly, they will die. And then we have a series of projects that we use that are all of this sort of flavor of trying to study individual cells in these tissues. The most common technique we use is one called patch clamp physiology. This is where we have a pulled glass pipette that you actually make a tight seal with a cell. You record its electrical activity in response to a current being injected into the cell. You can fill the cell with a dye, which allows you to look at the anatomy of the cell, like the picture I showed you earlier. And a modern take on this technique, which has been around for a long time, is to actually extract the contents of the cell. And you can sequence it using DNA sequencing technologies and then get a molecular analysis of that cell as well. So the sort of workhorse technology we have, this technique is called patch-seq, is to characterize using these three different ways at once cells in these tissue sections. In the course of trying to adopt more of the techniques that are available in model organisms, we've also developed a cell culture system where we can, or slice culture system where we can take these slices and keep them alive for several weeks in an incubator. And that actually allows us to use some of the tools of molecular genetics that are very common, working with a mouse, for example, to genetically manipulate those cells to introduce, say, at minimally a fluorescent protein into those cells so that we can label very specific types of cells. Or even we can do perturbations of various sorts. We can use all of the molecular genetic tricks that are available in a mouse. And so this is sort of really pushing the boundaries at the moment of being able to keep these alive for longer and to get much more specific about the types of studies that we're able to do, which really starts to bring many of the advantages of the tools developed in the models to be able to actually study the human. But those are the kind of main types of things that we're doing now. But anything that involves the study of individual cells or the connectivity among cells in this sort of local circuit can be done in these kinds of tissues now.
The thing I asked about that you haven't addressed is there anything novel or in the consent that patients are being asked to give to the points that she has raised at the moment, how you handle, or are the recipient beneficiary of this process? What about the consent?
So we have worked with the surgeons to modify their consents so that they are aware of the types of studies that their tissues would be used in. And then the consenting process itself, using that modified consent, is what the surgeons and their team discuss with the individual prior to the surgery to see whether they're willing to participate in that. And I must say the participation rate is extremely high for this. In general, these patients they would like to see something good come from this.
I would say that I've spoken a little bit with folks. I visited the Allen Institute to hear about how they are getting tissue and the distance between them and actually acquiring the tissue. I know that they've also reported that participants are very eager to do this. One aspect that I'm not clear on is whether or not, and this isn't always the case, but whether or not the treating physician should really be the consenting physician for research. Because there becomes an opportunity for what's called therapeutic misconception. And even with the clearest explanation, for all consent processes, people tend to not fully understand. That's just the weakness of informed consent. So when the surgeon who's going to be offering the treatment says, do you want to be in the study, it's hard to, one, deny that person the request, right? Because they're in this situation where they have treatment that you want them to offer you, or in a vulnerable position. And two, you might not understand that this isn't actually part of the treatment. So often what people recommend in this situation is that you do not have the healthcare provider offer consent for research. And it may be the case, I don't know, in your situation, that that's not the case. There might be a separate research team doing that. I don't know.
Okay, question over here, and then one in the front.
My name is Mikko Puttonen. I'm from Finnish newspaper Helsingin Sanomat, and I have two quick questions for you. One is I'd like to know how big are these specimens you have from the patients, and the other is more sort of philosophical problem with this brain tissue, because you could say that it's part of you take part of their mental life with with the specimens. So what because our mental life is created by the brain. So uh, uh, what do you think of this idea? And well, that's all.
So the first half of your question is easy. The typical piece that's resected is somewhere between the size of, say, a sugar cube, about a cubic centimeter, or maybe down to half of that. There is a rarely, the standard of care has moved increasingly to smaller and smaller resections. It used to be the case that these would be much larger. Most of these resections are now, quite small. I mean, I think it is of course, quite poignant that this used to be quite recently part of an individual's conscious thoughts and their memories. However, those are really distributed properties across a much larger circuit. The cells there, they make local connections. They also make predominantly longer range connections in part of this larger organ. So I think it's really a bit unclear what is the unit of consciousness, if that's sort of where you're going, and are we capturing that? My feeling is probably not. I think it's a bigger circuit phenomenon. I think, in this case, the question is, philosophical and less practical. These tissues are being removed anyway in order to treat the disorder. But I think we're not probably encapsulating very much of the consciousness in these very small pieces, personally. Maybe you feel differently.
Well, I thought that was very generous of you to say how poignant you thought his statement was, because I think that it is true that the whole reason why the field that I work in, neuroethics, exists is because there is something that seems to be culturally important about the brain, but it's identified with who we are. And I think you're asking a question about the size, because you're this is kind of a question of incrementalism. How much brain does it take before you freak out, basically? How much brain does it take before you're just, this is yuck, we can't do this anymore? And well, to some extent, that is a emotional response and an intuitive response. I think there's also a scientific aspect to this as well. So there are researchers who are trying to study consciousness and trying to understand even how to operationalize that in the lab. I mean, consciousness is a very complex word in philosophy. It means one thing, to scientists. Sometimes it just boils down to awareness, which I don't know if we would say is consciousness. But it seems like scientifically we understand that all of those cognitive experiences that we identify with ourselves require not only the whole brain, but immersion and exposure to a society around it. So that's kind of where, to stimuli, I guess, if you want to reduce it to something scientific around it. So I don't know that it would be wise to say at this point that a sugar cube size piece of the brain has consciousness in it. It doesn't, but one question does come is how, as Ed was pointing out, the reason we're creating these tissues, the reason why we're creating new models, is because non-human animal models often fall short as proxies of the human brain. So we have ourselves in an ethical conundrum. The closer approximation we get to the human brain, the more fidelity we have to that, the more ethically fraught it becomes, because it is like the brain in the human body. So I sit on the neuroethics working group of the U.S. Brain Initiative. And one of the things that we've been asking scientists to do is, as you develop these models that have higher accuracy, closer approximations of the human brain, are there things that we can take a look for to see if we might have any hints of it having some qualities that we might be worried about? So something that shows a certain kind of awareness. And another important question is, if these new preps develop that, would we even have the tool to measure it? And so that's another scientific question. So I really want to impress upon you that, you know, these philosophical questions can inform better science and aren't just a frivolous act. You know, and that's kind of how my colleagues and I operate in this space.
Question in the back, and then we'll come up front. Yeah.
This is a question for Ed from the web, from Nicholas Luco from El Mercurio in Santiago, Chile. He asks, could you apply the same methods you use to reveal the differences of mouse brain tissue and human brain tissue be applied to see if other organs of both mouse and human beings differ as much, especially when drugs are being used, presuming there is similarity between mouse and human being?
Yeah, it's a very good question. In fact, every other organ is far easier to tackle than the brain. All of these organs, or all of these methods are applicable to other organs. And so for example, the molecular methods that I mentioned are actively being applied to every organ in multiple species. There's a big multinational effort called the Human Cell Atlas that's actually trying to coordinate these activities to apply them to all different organs and together to build a map of the entire human body. Built in with that are applications to other organisms as well, to try to understand, you know, which actually model different aspects. And just like the pharmaceutical industry has found, different things will be better modeled by different organisms for sure. And so I think actually everyone recognizes that this is a very important thing forward to understand what could be a good model for each particular organ system and each particular disease that affects those systems, but they're very generalizable methods. Some methods don't really make any sense. The methods to measure electrical activity in cells are things to study neurons, so they're not really particularly applicable to kidney cells, let's say. But most of these methods are very, very generalizable, both by organ and by species.
Question up here in the front.
Hi, this is Rodrigo from Science Magazine. How expensive is it to do these procedures as compared to mouse studies or organoids? And is there any efforts to streamline guidelines or yeah, have some guidelines so other institutions can also have these programs?
I think one of the hopes of actually discussing these things is indeed to try to get more awareness that these approaches are important, and also that they can be expanded to other sites. Of course, a big challenge with what I've described is that it works with living tissue that has to be worked with locally. And so it hasn't really been possible to expand a single site to work with many different locations. However, any city where there's a hospital system that does these types of operations will be generating this type of tissue and could easily be used to establish a similar type of program. And I certainly have been active in trying to share our expertise so that could happen more easily. In terms of the costs, the costs once you're in the lab are very similar. It's a piece of tissue that you're doing various kinds of experiments on. The costs of working with surgeons are actually probably cheaper than maintaining lab mice colonies. So it's not an enormous cost for that type of access. The bigger challenge really is in establishing good connections between the researchers and the clinicians, and that's really an essential ongoing relationship that needs to be established for this to work. It has to be sort of a priority for both parties for this to happen well.
Okay, follow-up question here. Yeah.
This is another question. So maybe also on the ethical side. So once you have the tissue, how do you go about from starting from, just describing it, either the genome and also the architecture, and then kind of like playing with it as you do in optogenetics in mice or cell cultures. And is there I don't know if that's also the patient's consent to that, because you have all these tools and you can do so many things now. um, you said something about modifying them genetically for to label them. So how far can you go actually with these living tissues?
So at the moment, we have established this culture platform that I was describing that can keep these tissues alive for several weeks. They do eventually not maintain a robustness in that type of system, but this is actually kind of remarkable in of itself, that most tissue culture has traditionally been done in juvenile animal tissues because they're much more plastic, they can survive. That plasticity is also a problem because they change over time in culture. Adult brain tissues seem to be both robust and stable, actually. So they're kind of locked in place, and so they don't actually change their shape. They maintain their shape, they maintain their properties over time. The time is not indefinite, but it does give an extended period of time over which one can do these types of studies. The longer sort of trajectory of the research that I was describing is that by starting with a molecular definition of types of cells, one can then use that to look for regulatory sequences that are the reason that these cells have their identities, and use those as the driver for delivering the types of transgenes that can give you all those capabilities, like optogenetics, for example. All of those things are species agnostic, and so with a virus you can infect cells and you can turn on all of those types of tools. And so, we're really just at the cusp of this, but what we found is that this new type of molecular information we're able to get with these single cell studies gives you a direct path to be able to be very predictive about finding those regulatory sequences, and that then creates these tools that you can apply in all the different ways that you use in model organisms at the moment. So I would say this is really at its infancy, but compared to even just 5 years ago, there was no ability to do this kind of cell type targeting with the viral types of tools. And now it's a clear path to do that. So it opens up many more experimental possibilities in this span of time you can do with a culture.
Oh, question online, and then we'll come up here. Yeah.
This question is from Juwan Song from AAAS for Ed. Could you give a specific example of a key difference between mouse and human brains that you found from your study of living tissues? Perhaps one that might, with further work, impact the formulation of neurological drugs like SSRIs, MAOIs, etc.
It's a very specific question. So let me give two examples, because actually the specific thing that was asked is more from our postmortem studies. So as I mentioned, one of the things that we see when we're able to compare the same types of cells across species molecularly is that very often the usage of different genes is not the same between species. And one of the findings of our recent publication on this subject was that if you ask which families of genes are the most different between mouse and human, one of those turns out to be the serotonin receptors. So serotonin is, of course, involved in major depression. It's the target of SSRIs, and involved in anxiety and many other disorders. It turns out that the receptors that are turned on by by serotonin are among the worst conserved between mouse and human. So these are the kinds of things that we're seeing kind of routinely. That was one that actually popped out without looking, but you can take basically any gene family and compare them now, and you'll see many, many differences. There are also many similarities, but there are many differences. So, I think what that sort of illustrates is how you can't assume conservation. You really need this information to see whether this is a good model or not a good model. But now we are starting to be able to get that information. From the living tissues, it's a different sort of thing that's sort of coming out of that. We're able to see whether the properties of those types of cells are similar or different. And the answer turns out to be quite similar as well, that there are many similarities, but then there are really some profound differences as well. And those differences can manifest in a variety of ways. There can be more types of cells in particular cases. The properties of the cells can be really very divergent between species as well. For example, the firing properties can be really very different between a mouse and a human for particular kinds of cells. So, you know, it's difficult to name one because there are so many actually. And the question now is which of these are significant differences, such to the point that, you know, you would not want to use a model organism or would, but we're able to describe these things at this very fine level of granularity now with this in a way that has never been possible before.
Question in the front row.
I just wanted to ask whether this program at the Allen Institute here in Seattle is unique, or whether there are other cities or places that are using human brain tissue donations in the same way. Are you the pioneer and the only practitioner? I mean, you talked about wanting others to do it. Are they doing it?
Yeah. So we are neither the pioneers nor the only people to do this. There are a few other labs that I would really call the pioneers of this. There's the lab of Gabor Tamás in Szeged, Hungary, the lab of Hybe Mansfelder in Amsterdam, who have really established that this could be done. We've really built on this and expanded upon their pioneering efforts. What they really established is that the techniques that have been around and used in model organism tissues can be used in these types of tissues. What we've tried to do is to really standardize this, to build a bigger network, and to push the boundaries of the kinds of techniques that can be used with these types of tissues. But I would not want to take credit for that. We have started to have success at getting others to start to adopt these, and particular among younger researchers are starting to really pick up on this. And so I think it's a bit early to show this in the literature. But already they're getting, I can give you examples of new grants that are being awarded to start to do this. Lots of interest in trying to see whether this could be established, in particular at where the labs are very, very close to the hospitals, where it makes it even easier to do.
Karen, you talked about the importance of people being able to donate tissue if they want to. What are some of the major obstacles to people being able to donate? Or is it just awareness?
Well, it's not really a simple process to donate. You really need to go to their first, and maybe this is something you can speak to on a practical level, but there's not a uniform way in the U.S. really to donate your brain, period, much less brain tissue. There are really exceptional cases right now if you want to do the type of thing that Ed is doing. You already need to be part of a population who is slated for surgery, resective brain tissue surgery already. So those are kind of already limiting factors. For organoid type of research, it's a little bit different. I mean, because anyone basically with blood or skin can donate to those studies. And I'm not sure that if they're having problem with recruitment at this point, I don't know. I do know that just historically, that the reason actually I think I even got pulled into this conversation was because with the International Brain Initiative and 3 of the 7 international brain research projects are in East Asia. And several of the projects lamented that they weren't able to have brain banks. They were really having a hard time establishing them. And they were talking about whether, not just how to establish them, but whether it was even right to push people to donate. And it's still unclear totally what the reason is, but in part at least, it seems that there is a philosophical tradition, an East Asian philosophical one, that really prizes the brain as needing to be intact with the body. Your brain isn't even owned by you, it's owned by your family, and so you'd make different kinds of decisions about donating tissue. And I think those kinds of aspects are going to impact this type of research as you try to make it more widespread. And there are generational differences in these perspectives. I mean, in fact, even in the US, we have a similar sentiment. So I don't look to other cultures for opportunity to make a spectacle of the other, but for an opportunity to more deeply look at our own traditions and kind of the variation that we have within.
Could I just add to that? You know, even the post-mortem donations are very challenging. And in part that's because it's not our normal societal practice. People are organ donors, but that doesn't apply to the brain. And so it's really an onerous process to try to work with medical examiners to have access to autopsy specimens, which have to go through the whole process. There's no pre-consenting that happens in these, and it really dramatically limits what can be done with this type of research. It's a lot of effort to try to get adequate tissues to do this work, and it's often not very high quality by the time it's made it through that process.
I find that, just wanted to add that I found it very interesting to hear it put in this way, that the brain is not an organ in the sense for organ donation. That's why if you're an organ donor, it's not on your card. And the reason it's not an organ is because you can't currently transplant it. So until that changes, it's not really an organ, and you're in a whole other set of rules.
And once it does, maybe we'll think about it differently.
That's another talk.
That's a great note to end on. It's now 1:45. If you want to connect further with the speakers, they can be available in a follow-up room, 208 across the hall. But thank you so much for your insights today and your time. We appreciate it.